Hello, this is our video experiment for practical 1, spectrophotometry 1, protein analysis by subgroup 4.
My name is Roslana binti Rafael. My number matrix 106168.
Hello, my name is Moni Birit. My number matrix is 106045.
Hello, my name is Siti Nur Ayumi Piti Badaudin. My matrix number is 106395.
Hello, my name is Han Huixin, number matrix 104280.
Hi, my name is Cynthia Jialing. My matrix number is 103748.
The first material we use is the micropipette. This tool can accurately measure and transfer small volumes of liquid. Next, we use distilled water. It was used as a blank in the spectrophotometer. This means it helped to calibrate the spectrophotometer to ensure the readings are correct. Then, we had the Bradford reagent. This chemical is used to detect and measure the concentration of protein in our sample. We also use a BSA protein standard. This is a non-protein solution and it serves as a reference to help us create a standard curve. To make sure the liquid samples are mixed evenly, we use a vortex mixer. A cuvette was used to securely hold the liquid samples during the measurement in the spectrophotometer. We use a spectrophotometer to determine the concentration of a substance in a solution by measuring absorbance. We also use sterile pipette types to prevent contamination while transferring samples with the micropipette. Finally, we use test cube to place all the liquid needed for the experiment.
In part 1, a standard curve was prepared using BSA solution. First, 7 test tubes were labeled clearly. Using a micropipette, 4 ml of distilled water were added into each tube, followed by 1 ml of Bradford reagent. Then, BSA solution was added in the different volumes to create a range of concentrations. The first tube received 0 microliters and served as the blank. The second and third tubes received 50 microliters. The fourth and fifth tubes received 100 microliters, and the last 2 tubes received 200 microliters. And mix each solution thoroughly to ensure optimal interaction between the protein and Bradford reagent. Transfer 1 ml of each mixed solution into a cuvette. Next, the absorbance of each tube was measured at 595 nanometers using a spectrophotometer. The blank tube was used for calibration before measuring the rest. Record the absorbance readings for each sample. Calculate the average and standard deviation of the absorbance values and plot a standard protein graph with BSA concentration on the x-axis and absorbance on the y-axis at the regression line and standard deviation. In part 2, the same procedure was repeated for the unknown protein samples. 7 new test tubes were labeled. Each tube was filled with 4 ml of distilled water and 1 ml of Bradford reagent. Then, unknown samples were added in different volumes. 50 microliters of sample A were added to 2 tubes. 100 microliters of sample B were added to another 2, and 200 microliters of sample C were added to the last 2 tubes. 1 tube was used as a blank with 0 microliters of sample. Mix all solutions thoroughly and transfer 1 ml from each test tube into a cuvette. The absorbance of each unknown sample was measured at 595 nanometers with the blank used for calibration. Record the absorbance values for samples A, B, and C, and then compare the readings to the standard curve and calculate the protein concentrations using the regression equation.
Let's move on to the result of our experiment. So, this is our result for the experiment part 1. 50 microliters trial 1, 0.104. Trial 2, 0.116. 100 microliters trial 1, 0.127. Trial 2, 0.147. 200 microliters trial 1, 0.169. Trial 2, 0.198. As we can see, the absorbance increases as the volume of protein increases. Now, for experiment part 2, sample A trial 1, 0.146. Sample A trial 2, 0.137. Sample B trial 1, 0.152. Sample B for trial 2, 0.167. Sample C trial 1, 0.162. Sample C trial 2, 0.151. We can observe that part 2 also shows an increasing trend, except for sample C, which show a light decrease. Overall, this experiment shows that the spectrophotometric method is effective in determining the protein content in a sample. It supports the objective of the experiment, which is to understand the use of spectrophotometry in biological analysis both qualitatively and quantitatively.
First, our new learnings. We learned how to use the Bradford assay to analyze protein concentration and how to build a standard curve to estimate unknown samples. We also practiced using the spectrophotometer and understood how protein concentration relates to absorbance. Finally, we applied linear regression to interpret our results. Next, the challenges we faced. It was hard to pipette small volumes accurately, which affected our absorbance readings. In part 1, the BSA absorbance didn't increase perfectly with concentration, which make it harder to plot a clean standard curve. Calculating and applying standard deviation for error bars and setting up the Excel graph with a trend line also took some trial and error. Lastly, suggestions for improvement. We recommend adding a short pipetting practice before the lab. Also, providing a simple Excel guide or a pre-made template will make data analysis faster and easier.